We should go ahead and get started. Everybody who is an attendee, welcome. Thank you so much for taking the time out of your days to be a part of this. I think myself and I will let the panelists speak for themselves are really excited to have this conversation. I think we got a great group of experts that you're going to hear from. Unfortunately, Catherine Anderson wasn't able to make it. She did provide a couple of slides that if we have time, I'll go through, and I'll do my best to bring the story across. But I think between the group of people we have here today, we're going to have an amazing set of perspectives and conversations. So what we're going to try to do here is think about and talk about participant diversity from a pretty wide lens. So not just technological approaches, but processes and social approaches, and not just diversity in the narrow sense of representativeness and research, but also the structures that we build to deliver healthcare and research, and how those impact the diversity and representativeness, and ultimately the impactfulness of the research we conduct. So I just give a quick introduction to Sano and myself and why we're holding this webinar. So we're a platform for patient engagement in precision medicine research. So we have an online platform that allows people to get access to personalized reports and find research projects that are relevant for them. And we have at-home testing kits that enable at-home and virtual research. A big part of our goals as a company is to democratize access to research and build more bottom-up and patient-centric ways of running research that's accessible to everyone. So we've been working on developing the platform for the last 2 or 3 years, and we've been working with a number of different large-scale research institutions, including Genomics England. If you're interested in learning more about us, then feel free to visit our website or shoot either myself or Ross an email. So getting into the speakers and the agenda for today, what we're going to do is hear from 3 people, a couple minutes, around 5 minutes each, as an introduction to themselves and some of their viewpoints on the topic. As I mentioned before, Catherine can't join us, so our first speaker up today is Mavis, who's a postdoctoral researcher at Newcastle University. She told me she was nervous to go first, so she was going to be second, but I don't think she should be nervous at all, because she's got a great set of slides and an amazing perspective on this. So if you just give me a moment, I'll swap over to your slides, Mavis, and maybe while I'm doing that, you can introduce yourself.
Yeah, absolutely. Well, thank you for having me. This is exciting, and actually the nerves are, I think, a good thing, because it keeps me excited and thinking of my feet. So my name is Mavis. I am a postdoctoral research fellow at Newcastle University. I'm originally from Zimbabwe. And I've moved over not too, like, I'll say maybe 20 years or so ago, which seems to have shot past like that. And in London, I practiced as a midwife. So I've got that experience of working as a midwife for about 12 years in the wards, in community, but also as a research midwife recruiting women to biomedical research. And I've used that experience in understanding sort of how people navigate health systems, based as well on my anthropology. So I did a master's in medical anthropology, and this sort of helped me understand what we mean by health systems, what we mean by health and illness, and how these terms might actually mean different things to different people. And I'm currently exploring some of these issues, especially regarding research participation in Newcastle, and how we can make sure that the way we work also aligns to people's expectations. So that's where the ethics and governance comes in. Next slide, please. So I mentioned coming from Zimbabwe because, of course, we all have a way of looking at the world. And for me, this is based on me being a Black woman, a Zimbabwean, an African. All of these identities carry over into my work and actually influence the way I understand the world and how I interpret what goes on. And I explore some of these ideas when I did my PhD at King's, when I was looking at cultural identities and knowledge and research around familial cancers with Black and South Asian women. And this actually was a way to understand how people kind of worked through understandings of genetics, understanding of familial cancers, but also how they used those types of knowledge and interpretations to navigate the genetic system in the UK, and how they made their decisions to participate or not participate. And I bring all these influences, including the current work as a postdoc, to policy and practice in the projects that I do, which are both national and international. Looking, for example, at how we can talk about biotechnologies, how we can talk about genomics on a more global scale. And with this, I also take part in sort of the Genomic England Disparity Subgroup, where we explore some of these concerns. And on the next slide, you'll see I have a quotation which says, "Walking with the ghosts of those that have gone before us." And this is actually something that I saw on Twitter. Somebody had said, why don't people from different backgrounds participate in research or trust their doctors? And somebody had given this as a reason. I thought this was really interesting because it actually aligns with the way I understand and think about some of the underlying themes in my work. And I think it all comes back to context, that, everything that we do, the way we understand the world, the way we talk about ourselves and things that happen around us are always situated and connected. So we can't talk about research practice without talking about, biomedical knowledge. We can't talk about practitioners without talking about patients and publics. And so we have to always be thinking about how all of these things are connected. And this is important because when we look at the next slide, we have to go beyond thinking of maybe, let's say, genomics as something that is a one company thing or a one country thing, because what you end up with is on that bottom set of on the pie charts, is this great disparity in the people who currently take part in genomic research or who are in sequencing data. And what you see at the top is, when I mentioned that we have to think about practitioners, we talk about genomics as a global thing, but actually when you look at who's participating in that conversation, there's a very big difference in, for example, genetic counselors across the world. So we have to consider how our work can impact people, but not only as patients, but also, in terms of who's taking part in that conversation. So yeah, lots to talk about. I'm excited for this, and happy to connect afterwards if anyone's interested as well.
Great. Thank you so much, Mavis. Wonderful. I am hopefully going to transition well here. So our next panelist is Charles Steward from Congenica. Really excited to hear his perspective. He has a background in genomics research and is also a parent of 2 children affected by rare disease. So Charles, I'll hand the floor over to you now, and I can operate the slides.
Yeah, look, thank you, Patrick, and thank you for inviting me to speak today. That was a great introduction from Mavis. Not sure I can better that at all. So anyway, next slide, please. So yeah, a little bit about me. So I'm the Patient Advocacy Engagement Lead at Congenica on the Wellcome Genome Campus in the United Kingdom. And I've, next point, sorry, just hit return. So yeah, I spent around 26.5 years working on the human genome, which of course has given me a huge understanding, I suppose, of the different technicalities and challenges that we've had to overcome during those time. And actually, when we started working on the human genome, we were given a sort of a period of around 50 years to get it done. And that was back in sort of 94, 95. And it's astonishing to think now that it's possible, and this is a very cherry-picked example. I mean, this is really very streamlined, but it's possible to take the blood from a patient, sequence it, analyze it, and get a genetic report in 19.5 hours. Now, admittedly, that's a Guinness World Record for Rady Children's Hospital in San Diego. But that just shows in those 26 years how much progress we've made in being able to understand and interpret the human genome. So next point, please. So part of my career was involved, obviously, in working on the human genome, but more specifically, I was interested in understanding what the genes look like within the human genome and what they actually function for. And part of that was me leading the initial human chromosome 10 annotation and analysis that was published in Nature back in 2004. Next point, please. So actually, so I did my PhD in molecular genomics at the Wellcome Sanger Institute. And the Wellcome Sanger Institute, which is also on the Wellcome Genome campus, is most famous for generating the largest proportion of the human genome, which is 1/3, more than any other institute in the world. And the human genome, which was actually published 20 years ago, last month, 26th of June, I believe. That was when the draft human genome was done. Next point, please. So I spent quite a lot of my career doing teaching, and I think of particular interest to today's discussion about diversity is the work I did for the Wellcome Trust up until about 2016, where we, a sort of a team of 6 of us, were sent to different Wellcome Trust outposts around the world, typically to areas of the world which had less experience with the human genome, where we would go out to Africa or South America, or even sort of Southeast Asia, and help people to understand what the human genome is and all the different public available sources and analysis software that's available. And then I'm also a lecturer for the NHS Genomic Medicine Master's degree. And this is I think this is a really great thing to come out of sort of the whole GEL 100,000 Genomes project, where healthcare workers, it could be anyone from sort of a nurse to a consultant or what have you, are able to go on these courses and learn what it is, what the human genome is and what it can do and why it's so important. So I think this is a really important point for discussion. You know, talking about diversity, we need to talk about diversity of opinion, where medics, I think, and scientists have probably traditionally struggled to sort of bridge the gap, which is clearly closing now, thanks to these sorts of initiatives. Next point, please. So particular interest to me, and this is based upon personal experience, I've done a lot of work looking at the genes that involve with severe and early onset epilepsies in children, or often, you know, sort of in babies or young infants. And that's driven by my daughter having a really nasty epilepsy when she was around sort of 6 months old. Next point, please. Yes, so also, outside of my everyday job, I'm a member of the 100,000 Genomes Project patient participant panel. So I'm sure all of you are aware of the 100,000 Genomes Project. Now that's fine, you can move on to the next slide.
Sorry.
This is all really relevant here. So I'm sure you're all aware of the 100,000 Genomes Project. It does what it says, you know, it's looking at the genomes of people with rare diseases and cancer. And part of the initial sort of setup was to make sure that the patient was at the heart of the project. So when the project started, people were invited to join the panel, and we have direct influence, direct input on everything that occurs for the 100,000 genomes. And that can be from, for example, you know, Patrick came and spoke to the participant panel to say, I would like to do this project. What do you think? You know, and the patients, the participants, then sort of had a rubber stamp of a seal of approval. So I think this is an absolutely essential and one of the cleverest ideas, so simple in many ways, but having the patient at the heart of the 100,000 genomes. Because, and I've experienced this before in my academic work, you know, people think, oh, this is a really, really good idea, let's do this. And they build a fantastic piece of software which does, you know, amazing things, but when you want to use it for your actual job, it's absolutely useless. So, you know, the patients decide what we think is essential outcomes for those people on the project. So as alluded to, this is me, and that's my wife, and my 2 children, Imogen, who is going to be 8 in 2 weeks, and my little boy Jasper, who is 2.5. And both of them were born prematurely. Imogen at 30 weeks, she developed fine for about a month. No, sorry, she was in intensive care for a month. But then after about 6 months, she sort of regressed in her development. It's really quite worrying to see. And then after sort of a month of that, she began to develop these really nasty sort of jerks. And then she was diagnosed at Addenbrooke's Hospital in Cambridge with something called West syndrome, which is a really nasty catastrophic form of epilepsy. And if you could control the seizures, then you give the child a chance to develop again. But of course, every time this child has a seizure, each seizure causes more regression. So if you don't get control of the seizures, unfortunately, sometimes it can result in early death. So Imogen was enrolled onto the Deciphering Developmental Disorders, which involved looking at the exons or the bits of the genome that are responsible for producing proteins, and also looking at arrays CGH. So looking at sort of large structural variants in our genomes. And it was a trio, so they could look at mum, dad, and her. Nothing was found, unfortunately. So she was then put onto the 100,000 genomes, which actually looks at the whole of the genome, although realistically speaking, we're probably looking at similar sorts of amounts of the DDD. Nothing was found there. Then my son was born prematurely, and unfortunately he suffered very severe brain damage during birth, similar to my daughter. And he was also then put onto a genome study at Addenbrooke's, and nothing was found. So next slide, please. Does that mean that it's not genomic? Does it mean it's worth having a genome studied? Even now we know we haven't found anything. Does it mean it's been a waste of time? Well, absolutely not, no. It means that we know it's not one of the really nasty disorders that clinicians know about. And I think it's interesting that if you look at the 100,000 genomes, it was set up by our ex-Prime Minister David Cameron, and ultimately he was inspired by his own son's battle with probably the most catastrophic form of epilepsy called Ohtahara syndrome, which is very similar to the type of epilepsy that my daughter had. And sadly, he succumbed around the age of 6, I believe it was. So, rare diseases, genetic disorders, terrible things like this, they touch everyone. They cross the whole spectrum of the global population. Thanks.
Great, thank you so much. I will close out here. Stop sharing my screen. Thank you so much, Charles. Very, very much appreciate the overview. So our final speaker is Jessica Mai Sims, who is going to introduce herself without slides, so everyone can focus on the words she's saying. Jessica, thanks so much, and I'll turn it over to you.
Thanks, Patrick. Yeah, sorry, the lack of slides. Just as an introduction to myself, I've just returned from maternity leave. This is my first official duty since coming back, and so really pleased to be here. Thank you for having me. So again, my name's Jessica Sims. I work for the UK CRC Tissue Directory and Coordination Centre, and in short, we're a project to support and improve biobanking nationally. And we primarily do this through our tissue directory. So it's an online directory free from the point of use. And it's targeted primarily at researchers. So the idea that, let's say I'm a researcher, I need some samples, I'm having problems recruiting for my study. But what I might know is that there are places all over the UK that already have existing samples in fridges or freezers that I could do my research with. So it's about going onto a directory, entering my disease area, identifying what kind of sample sets I would need to do my research, and then getting in touch with biobanks, biorepositories, clinical trials, cohort studies, anybody that might have these samples that I can use. And also we have a data directory, which is newer, and it combines a number of cohort studies. So if you're interested in answering some more of the data queries, so perhaps you don't need samples, the information is there as well. And so I'm one of the engagement leads for this project. And I think unusually for biobanking, my background is in social sciences, and specifically race equality and patient and public involvement. So I've done work on ethnic inequalities in different policy areas at the think tank, the Runnymede Trust, health inequalities in relation to organ donation and transplant at King's College London, and even some marketing and advertising work for non-white groups. And so throughout my career, I've been very concerned about who is being researched and why. But however, the attraction to patient and public involvement is that it's been grounded in the idea for research application success, different groups need to be involved. And, Mavis and Charles, they've already touched on this. And I know for this webinar, we've got some really big questions, undoubtedly our audience want answers to. However, I'm just going to take a step back. So if we take for granted that we want diverse research participation, how do we get there? So engagement is important. So this idea of explaining research, raising awareness about it in ways that are tailored to different groups and how they prefer to receive information, where they go, what they read, who they trust. So that's about engagement. Back into involvement, having diverse involvement in the research life cycle. So who's doing the research, the testing, the evaluating? Who's getting the word out to those places where others go for information? And a very general example, and so in a bit other, say you want to do some research on a surgical intervention. So one of my career backgrounds is I used to work for the National Institute for Health and Care Excellence, NICE, on surgical intervention guidelines. And we'd see different products a lot, and we'd assess the safety and efficacy, but mostly just kind of if it's going to work, and they haven't been approved yet. Okay, so say you've got a widget and it's designed to improve your treatment and your care, and you come up with this great idea, this great product, and you realize that this product is really, really great, but it's great for people like you, and you're 6 foot tall, 100-pound man, and maybe less great for 50% of the population, so women who might have a smaller frame. And we're just talking about here in the UK, what about the usefulness of this application in other countries or continents where your company might want to market to? So you find this out because your research participation is diverse. That's great. But maybe it would have been flagged up sooner if the different types of people who had been involved in that research at earlier stages had offered their input, their life experience, so on. We've already heard from Mavis and Charles about how, them as people influence what they do, what they're interested in, how they approach problems. And that goes for all of us, really. And so it's about the recognizing that there's different ways that we can get people involved that then improve the participation. That's it.
Wonderful. Thanks so much. I think we've got a couple of threads coming together here. For those of you all who are listening, please feel free to use the Q&A functionality or the chat at the bottom. So I've, as you all know, well, you may not know, we have a podcast and I'm the host, and I'm used to asking questions for an hour straight. So if we don't get any, then I'll absolutely do that. But I'd much prefer if you all came with some of your questions. So I've got a couple just right off the back of this, but please feel free to click on the Q&A button. It should be across the bottom of your screen. I'd like to just pick up on that point that you just made, Jessica, which is flagging up potential issues much earlier on in the process. And I think this relates to some of the points that Charles and Mavis made as well. And I just wanted to put this question to all three of you. What are the solutions that you see to do this involvement of patients or other stakeholders earlier on? And I'm really interested in exploring what is technology and what is non-technology, because I think often we kind of fall in love with the idea that there is always a technological solution, but sometimes it's not necessarily just about technology, or maybe the technology is enabling, and it's really more about who's involved or some element of the process. So I'd be interested if any of you all have thoughts on that, how we get people and other stakeholders involved earlier on in the process.
So if I speak from my patient participant panel involvement, we had one of our first, I think it's the first or second meeting, and we were all sitting in the room, and we were talking, or we were being talked to about all the different processes that were going on for the 100,000 genomes, and it would be sequenced and you'd be analysed, and then eventually you get a report back if you're lucky or negative, have you? And we asked, well, that's great, but some of us have had experiences before where we've had blood samples taken and they've just disappeared. They've gone into someone's fridge in a lab and they've never been seen again. So how can you guarantee that the 100,000 genomes won't make the same sort of mistakes? Why can't we have, it's a bit like a sort of a DHL track my sample sort of idea. And this wasn't something that they'd actually considered, because while obviously it was possible for probably a bioinformatician to check exactly where that sample was, there was no easy way of doing that. So this, I think, was one of sort of bringing the patients together and getting them to talk about what was really essential for them, and perhaps had been overlooked by the people who set the project up. This was, it didn't mean that you got your samples any quicker. It just meant sort of peace of mind. Your sample was trackable, which meant it hadn't been lost.
Yeah, so that's really interesting. I like the idea of bringing people in from the beginning that Charles was talking about. And for me, I think it doesn't always have to be a technological solution. It starts right from the beginning in designing your research question. What is it that you want to find out, and who are the people that you need to help you ask that question? In defining your population, yes, we talk about diversity, but why that particular group of people, and why are you defining that group in the way that you are? Because sometimes you go out and you say, I want to talk to people who have this skin condition. There's a brilliant book that's coming out, I think, later this year called Mind the Gap, and it's by a medical student. And this is something that has been talked about, for quite a while, about people presenting with skin conditions, but actually maybe the presentation is not what medical students have been used to or taught. And so what this book does, it just has photos of, dark-skinned patients and how these conditions actually show. And something as simple as that of just saying, well, actually, it's not necessarily research, but it's just already tapping into the knowledge that we have. So when you then want to do research, you say, well, what do we already know in this area? Think again of your history, think of the context. Define your population according to your aims. And Jessica talked about engagement. Make sure that the way that you interact with people aligns with the expectations that they have, and you can only do that by asking, well, what's important to you? So as researchers, we might have things that we think are amazing questions to ask, but unless you ask the people who might be impacted, you don't actually know if that's a useful thing or not. And I think technology is great, but like Charles and Jessica already said, without understanding people's needs, you could have this amazing piece of work that isn't going to work in practice.
Yes. Jessica, I was wondering on Charles's point about tracking the sample through the 100,000 genomes process. I imagine you probably have similar discussions in terms of the UK tissue repositories that you're part of. It's probably much more challenging and on a much more complex scale, but have you all had any discussions around whether participants in this resource might like to see where their samples having an impact in research, or or maybe it's something that already exists in some form?
Well, yes, this has come up, and part of the challenge that we have to deal with is that patients who are giving their sample, donating their data, they want to know what research has been done, what is the impact. However, the problem with research reporting is that when they go into the system and they're anonymized in some way, that's where that thread just kind of breaks down, and you can't really see what has my involvement been, or my participation, what has that exactly led to, because there's no way you can really trace that. The best that you can do is keep the people that have been, they've donated their time or themselves to your project, is really keep them in the loop. And so often what's talked about is how important research dissemination is. So this idea that, we publish papers, we might publish papers a few years after the study has happened, and then it kind of goes off and we're involved in some other project. But really what patients want to see is like, I did this. whatever happened to it? And then it requires them to go back to the researcher who may have left, or back to the project, and find out really what's happened. So it's about kind of thinking about research as not just what the researcher is doing at that exact moment, but really thinking about the legacy of it. And if we are really committed on ensuring that people are involved, they're listened to, they are valued, research dissemination it is really, really important.
Yeah, thanks very much. We've had a couple of great questions come through the Q&A. One that I think is directly relevant to this is from Amir Gandar, which is, can the panel give some examples as to how to effectively engage different groups at an early research stage, particularly owing to researchers being predominantly trained to design and conduct research studies or come up with hypotheses, but not necessarily the practical knowledge in how to actually engage. I wonder if you all have any examples of how this has been done effectively.
So I can start this one off. I mean, I come from a situation where I work in a university, and I'm very privileged in the fact that we have structures that support researchers to do something like this. And so you may have colleagues that already have set up kind of participant panels, or have involved and connected with, say, research ethic committees who have patient involvement on their committees. And so we have structures built in and resources that one can use to tap into. The difficulty being is knowing where to look. And some places do this better than others. And once you get into this rabbit hole almost of patient and public involvement, you find that there are loads of resources, and there are people who this is their life, this is what they want to do, and they've made it their job to help researchers. And so they have the ideas. Nobody can be an expert on everything. If I'm a researcher and I know my research back to front, okay, but that doesn't necessarily mean that I need to also be a marketeer. I don't need to be a whiz at social media. It's about connecting to others that are clued into different community groups with different ways of communicating, and asking them, what would you do? And how can you help me to make my research or my process better?
Charles, do you think there should be a participant panel in every research project?
Yeah.
What do you think?
Absolutely. I mean, it's worked so well for the 100,000 genomes. And I think the fact, one of the key things is we've got enough people to make it work, but not so many people that no one really is ever able to say anything. So I think, yeah, the participant panel, I mean, that was great what Jessica said, that it's essential. And being at the heart of any project, being able to sort of have clarity over absolutely everything that's going on, and as I said, sort of like a final say. But I think sort of expanding on that idea of not everybody knowing everything, I agree, that's absolutely true as well. And I think having been involved in teaching for the genomic medicine course for the NHS, I don't think we can ever expect our clinicians to be world-leading geneticists, although clearly there are some, but I'm talking about sort of day-to-day people who work in clinics and see patients, like people like GPs. I think what is important, though, is that they have at least a sort of an understanding of genomics, and perhaps the ability to tell a patient what's important about their genetic disease, rather than telling them, this is the human genome, this is how many chromosomes, this is how long it is. That's not important. I think the important thing is to be able to sort of pull out that little bit of information that will make the patient's life more comfortable, more interesting, or more understandable. So yeah, that absolutely.
Great. We had another great question come through the Q&A. There's also a follow-up to one of the points that Jessica was making earlier, which we can get to in a second. The question on the Q&A was from Hannah Thompson. Do we really think diversity in biomedical research is possible when so many of our social structures are still inherently racist? And a similar question for precision medicine. So that was for Mavis specifically. And then for precision medicine more generally, do we think that precision medicine can be available for all when we don't yet have social equity, sorry. Any thoughts on that?
I absolutely love that question, because I think it's very true that we cannot expect diversity to be this amazing thing in biomedicine when actually we're ignoring the social structures. And I think the two actually go hand in hand. That medicine and medical research cannot exist in a bubble, because actually when we go back to thinking about context and how people interpret the world, it is also based on historical colonialist, racist ways of working that have, that people remember from their parents or their grandparents. And so until we acknowledge those harms, we just have to look at what happens in African countries. We have to look at what happens with indigenous populations. All the concerns that they have are somehow tied to the practices that have happened, which continue to have a legacy now. So I think, yes, we can talk about, diversifying biomedicine in all these amazing ways, but actually we can also be advocates, and we have to use that as, as a way to be activists for people's lives. Because how can we say, yes, we give you this, answer to this medical problem, but actually you still live in a place where you don't have access to the very same medicine because you don't have the right insurance, or you don't have access to clean air, clean water. I think the two go hand in hand. And ideally, we should bring all practitioners together, social scientists and medical practitioners and researchers, to try and tackle these, instead of, letting precision medicine or biomedicine remain in an ivory tower. So it's possible, but not by itself. And I think that's a really great thing that we all have to be considering, because social justice is part of medical practice, or it should be at the very least.
Absolutely. Charles or Jessica, any thoughts on that?
Well,
yeah, Jessica, you go first.
I absolutely agree with Mavis. I mean, what I was going to say to that question is that, yes, biomedical research can definitely be diverse. You can have diversity, but can it still be racist within that diversity? Absolutely, yes. So one of the things that I'm more concerned with is this idea, okay, yes, diversity, but what Mavis said was about social justice. It's about equality or even equity and social justice. And so is this research that we are conducting and that we're delivering, is it just, is it appropriate? Does it provide value and meaningful value? Not just having different people in a room, but whether their opinions are valued or whether their experiences are reflected. Not that, oh, this is an outlier, so why don't we just get rid of this right now and let's focus on the group? Because what I'm interested in is why is that person or why that population group is an outlier, and what can we learn from that? And it's only until we reach that point do we derive all societal benefit and justice.
Absolutely. Charles, were you going to add something to that?
Yes, I mean, it was only sort of last week that I was reading about this large epilepsy genome-wide association study that was done in Japan. And of course, realizing that most of these sort of genome-wide association studies that are carried out are typically Eurocentric. They're based upon European populations. And this strikes me as pretty awful, really, because if we want to go into, say we want to start up an epilepsy project in Africa, and we do some exomes or some genomes, we're really, really restricted by the fact that, as far as I'm aware, there isn't a control population. So that, there haven't been enough studies done on sort of the African population such that we can actually derive meaningful information about sort of rare variants and how we can sort of filter out the variants in a genome that are non-contributory towards sort of pathogenic disease or pathogenic variants. And so it's certainly a huge need to invest in sequencing in these areas. And another astonishing stat I was reading about, because of course I've got a big interest in preterm births, that in the UK, if you're born at 24 weeks, so that's sort of 16 weeks before term, you have a 50% chance of survival. Now in Africa, you only have a 50% chance of survival when you're born at 34 weeks. Now think about that, that's 10 weeks difference. which is obviously, you know, that's you know, clearly down to the ability of sort of the medical care there. But what I do believe is, you know, one of the really good things that's come out of sort of technology in the past few years is the internet. You know, if you have an internet connection, you are able to access, you know, many analysis products if you're a clinician or a scientist across the internet for free. And I do believe that, you know, potentially because of these sort of, you know, clinical geneticists or clinical scientists who are, you know, really specialized people who, as we saw from Mavis's slide, you know, they're centered in United States and Europe. That you're able to actually, through the internet, to access these people's specialities. So, you know, you begin to sort of spread a bit more equity around the globe through the use of the internet.
Yeah, absolutely. And I'd love to pick up on that thread of virtualization and digital tools. One of the, you know, what Catherine was going to touch on was some of their work around virtual clinical trials. And in June of last year, the FDA in the US, obviously the body that reviews new medicines and is responsible for oversight of clinical trials, announced that they were going to be taking a close look at using virtual clinical trials or recommending more widely adaptive trials, virtual trials, and ways to level the playing field, because there's been historically a lot of data that indicates that people from BAME backgrounds end up incurring greater costs to participate in clinical trials and participate at a much lower rate than the representation in the population. And there's obviously a huge number of factors here. So I was interested in any thoughts that you all had on how technology can play a role, whether it's virtual clinical trials or ways of engaging communities online through community boards and those sorts of things, or other ideas you had. A lot of the people in the audience are either involved in clinical research or in large scale research programs and looking at solutions they can use to make sure they're reaching the widest audience. So I think they'd appreciate your views on that.
So I'll weigh in a little bit. Well, and forgive me, I'll be thinking as I talk, so hopefully I'll be clear. I think it's great to open up as wide as possible the use of tech to enroll people. We also have to remember that not everyone's going to have access to internet. Even within the UK, not everyone has access to internet. So you have to also think, well, okay, so who am I reaching with this technology? Is it going to be educated people across different ethnicities? Is it going to be people who are only interested in either the disease or taking part in research? What are the other ways that you can then use that tech? Maybe go to social media, for example. Twitter has a very big minority presence, like young Black people are on Twitter a lot. Find out how you can talk to them about some of the concerns they might have for that technology. But I think also in relation, we have to think about how the use of technology sometimes also exacerbates the inequalities when you think about big data, right? So it is, yes, using technology to enroll people, but also we have to figure out how technology has also been used as surveillance tools that will maybe stop people from enrolling or participating as citizens or as members of the public. And we have to, again, kind of think beyond just the enrollment of people, but what are these tools? How are they used just in everyday lives? And can we turn that maybe negativity into something that people will want to be involved in? Local radios, going out into people, like you can never overestimate the power of just having a conversation with someone, and people can actually see you and talk to you and ask questions. I think that's, yeah.
Jessica, I know you've published some work on gamification and using more maybe technology that's relatively foreign to most research. I wonder if you have any thoughts on or learnings from that you can share.
Yeah, the first one I was going to say on that point was that what we know about certain groups. So my background is kind of understanding different ethnic groups and how they participate in society, in UK society. So I'll speak from that aspect of diversity. We know that it's a younger population, ethnic minorities, younger population. We also know that they are early adopters in terms of technology. And so if you wanted to engage people with technology, the data says that these are the people that should be your early adopters. And so there's kind of an idea that, well, if they aren't, there might be something wrong with your approach or wrong with your tool, or, you know, what is that? What needs to be investigated, which means that you're not reaching them for some reason or another. And so some of the work that I've done is about games, because I'm, you know, interested in engagement in research. I'm also interested in using creative methods. So this idea that, especially in academic research, I'm not going to have lecture and get in members of the public interested in my work just because I'm going to lecture at them. That's going to really only speak to a very specific segment of the population. So this kind of goes back with this idea of technology. It's about market segmentation. It's understanding what your audience is. And it's not just the public or young people or whatever. It's about how specific can you get that audience so you really know that what your message is going, it's very tailored, or the method that you're using, it's very tailored. So for example, I've made games on transplantation and biobanking, and I specifically chose to use board games. So not digital games, because I wasn't interested in digital games, because personally I'm interested in, you know, analog board games. So I was trying to speak to my own experience of what I like to do and find a target group of, oh, there's all of these people out there that like a very specific thing. It's in some ways, it's kind of niche, adults that play board games. So not children's games, but, you know, very strategic, highly cerebral, involves a lot of planning. Oh, that's also kind of like my job. It's kind of like biomedical research. So the topic area, the medium, and then I had my message, which was raising awareness about either transplantation and organ matching among ethnic groups, or about just how samples are collected and used in research. Made something, went out, taught people it. If I gave them a lecture, they wouldn't have come, you know?
Absolutely. Yeah, I love that. That's amazing. Charles, do you have anything to add to that? We have another question come through the Q&A after you've had a chance to weigh in.
Nothing as technical as what Jessica's just said. What I can say about social media actually has played a huge part in many people who are involved in rare disease, because, you know, social media, Facebook, it's a way of connecting with people who have the same rare disease, you know, all around the world. You know, 20 years ago, you were given a genetic diagnosis, and often these are terrible genetic diagnoses, but you would have been on your own. You would have had to live with your diagnosis without really engaging with anybody else who has the same thing. But now, so with Facebook and Twitter, you know, there are specific groups set up based upon a specific mutation in a specific gene. And you can draw strength from each other. You can share ideas. You know, you can speak to someone who's right the other side of the world and say, well, what do you do over there? And we do it like this here. And you can share information and take strength from each other. And, you know, it's interesting, the power of the patient. You know, the patients are actually able to drive research. You know, they can club together, they can raise funds. And it's amazing the number of patient groups, you know, for these terrible disorders that people have, are actually driving the agenda for pharma. You know, pharma are taking a huge interest now in these patient groups. So yeah, from that's what I have to offer for that.
Yeah, we had our first live podcast last week or the week before with a guy named Keith MacArthur, who founded CureGrin, and you'll know Gillian Hastings Ward, who's part of the Genomics England 100,000 Genomes program. And I just think it's amazing how there are these patient-powered research networks that are completely flipping the script. And I'm interested in as well, whether it's possible to do that in communities. You know, it doesn't necessarily need to be based around, you know, a rare disease necessarily. It could be based around a local community or otherwise. Yeah.
Yeah, absolutely.
We had a great question come through from Kevin Garwood. Could you please comment on how you think the state of diverse patient engagement varies across different countries? And I'd like to add to that as well, if there are any specific places that are doing it really well, either research programs or or approaches, I think it would be really useful to have some of those positive examples.
So there's different ways of looking at this, right? You can think of maybe principles. So instead of just going out to people, think of what principles might actually unite people. Places like the NIH are involved, have put out a set of ways of thinking about how to engage with different publics. Places like The Voice, which is actually based in Newcastle, have ways that you can reach out to different participants and think a little bit more about how to get them into research. But actually, I think there's also something to be learned about, I mentioned early Indigenous populations, going out to those communities and saying, well, we have this piece of work that we want to do. How does that align with the values that you have as an Indigenous community? You can look at work in Canada, Australia, in South Africa, where researchers have actually had to think differently. And I'm talking here not so much diversity in terms of people, but in terms of working practices, where they haven't just gone and done helicopter research or where they just take samples and go away, but actually saying, what is it that people need? And one of my favorite examples is not really biomedical research, but it's about the rooibos tea in South Africa, where over years there was local knowledge that then got sort of industrialized, and how those communities came together. The San people and the Khoi in South Africa came together to work with industry to say, this is what we need from you to recognize the knowledge that we have, but also to advance the knowledge that you will have with technological advancements. And then the creation of the rooibos, I can't remember the full name, but it's sort of like a benefit sharing. So I think it's not always about just getting out to people, but really thinking in terms of values and principles, and underlies the reasons that you want to talk to people and engage with people. So not really a specific example, but definitely ways that we can learn from how Indigenous populations approach things like genomics, how research is done in African countries, engaging community leaders, engaging different people who live in rural areas, and bringing them in and saying, what is important to you and how can we think about the benefits of you participating in research.
Excellent. Anything else to add on that? Great. So to close off here, I was wondering if each of you all could just give your thoughts on, to anyone thinking about starting a new research program or project and wanting to make sure they get it right from the start. What's one or two critical things that people need to do at the earliest possible stages to ensure success, or at least get close to success?
Well, you know what I'm going to say. Have the patient at the center of everything. Involve the patient as soon as you can, absolutely as soon as you can.
Mavis, Jessica?
Work with people who are different to you, because they will just bring different perspectives, and be prepared to be challenged. And whether that's research colleagues, clinicians, patients, but try and get people who look different or think differently to you to have these discussions as you design your work.
Excellent.
Well, this comes with some caveats depending on who you are and who your family is, but and what your research is. But how could I explain what I'm doing to my mother? Like, if what I've written down, if she can't understand, or maybe she does understand, but has some questions. And so the idea that it's not just about going out and thinking about who all of these other people in the world, how they're going to react to whatever it is you're doing, but think about the people that are closest to you that actually already care. So they already have a shred of interest. And so they'll be empathetic, and they'll want to learn more. And so how would I explain it to that person? And how would I make sure that they're on board and they remain interested and they care about what I am doing?
Great. No, thank you all very much. Just see if we have one quick question that's come through. How important do you think it is for students as early as high school to know about and get into research and gain experience on diversity and impact of biomedical research? Is there something we could do earlier on in education?
I think it's hugely important because from a very early age, you know, medicine impacts us in everything that we do. And trying to make people realize, I think we need to teach at an early age the reasons that we need to talk about cultural diversity and different ways of thinking. It's not something that you do only when you get to, you know, university, but if you embed understanding difference from an early age, it doesn't then become, oh my goodness, I should have known this. Is like, this is just a natural progression from when I was a child and valuing people's different opinions.
Yeah.
Absolutely. Great. I'm just going to close out here with one slide, just to let you all know what's next on this. So thanks so much, first of all, to the speakers and attendees. It's been really great. I certainly learned a lot and appreciate all of your time preparing for this, bring your expertise, and also to the attendees for the great questions. For the attendees, you will receive a brief feedback survey. We'd really appreciate it if you could take some time to fill that out so we can hold more of these great discussions. We'll also follow up with an email summary of some of the highlights and the video as well. If you have any ideas for future topics, please just let us know. I'll also shamelessly plug our podcast where we'll be continuing the conversation there. So we have a really exciting episode that just recently recorded with Daphne Marchenko that will be coming out in a few weeks, which is focused around the topics of genetics in education and some of the issues of the way that genetic testing is starting to be used in education. And we've had some past episodes that covered Eurocentricity and genomics. So if there's anything you need from us, then please don't hesitate to contact us. It's email at contact@sanogenetics.com. So thank you again to the speakers and attendees. Carol Scrafton has said thank you in the chat. So thank you all very much, and have a great rest of the day.
Thank you.